Ooh. All right. So, what I've done here is I've just drawn 2 neurons in sequence. Okay, there's one neuron here, and there's another neuron here. And essentially, in this diagram, the flow of information is going, so let's say, coming from the environment to this part of the neuron. It's going to flow in this direction and be transmitted to the next neuron. So I'll just say flow of information. Okay, it's just flow of information and talking about it. And this is my other favorite stuffy. This is my.
It's a lobster.
No, it's a neuron. It's a neuron stuffy. Okay, and I like to use him to illustrate some points. So, how many neurons in sequence does it take for my finger to touch the stuffy, to feel the stuffy, and then transmit the feel of that stuffy to my brain? How many neurons does it take to do that? Do you know? In sequence, just a good number. Well, it is a good. I guess it's a good number. I don't know. Any guesses? 3.
Only 3?
Just 3. It's a 3-neuron system. Okay, we'll get to exactly what all those are as time goes on. But essentially just 3 neurons. Okay, there's first-order neuron, second-order neuron, and third-order neuron. There's somatosensory, and we're going to learn about that when we get to spinal cord. That's next week. We'll worry about that next week. But essentially, it's a 3-neuron system. Okay, when you look at this neuron, now that's for somatosensory. How about for motor? How many neurons in sequence does it take for to go from my precentral gyrus all the way to my knee, let's say? How many neurons is that? I see 1. I see 2. Anybody else? There are 4. The answer is 2. 2 neurons. And the ones that we're going to learn about this year and talk a whole lot more about next year are upper motor neurons and lower motor neurons. That's it. Those are the 2 that you have to know. So realize Spencer has a really long lower motor neuron going from his spinal cord all the way down to his big toe. That's just 1 neuron, right? It's the axon of that neuron. The cell body lives someplace else. We'll talk about that. But these are big structures, right? Large, very long. Okay, so 3-neuron system for somatosensory, 2-neuron system for motor neurons. In general, the neuron itself has a couple of basic parts. My stuffy is a standard multipolar neuron. Okay, multipolar. It has this cell body here, or soma. That's where his eyes are. And then he's got dendrites on the outside. He's got an axon. Okay, and then this is going to be the axon terminal. Okay, synaptic terminal down here. He also has some rings. That's intended to designate that there's a special fatty coating on the outside. Does anybody know what that's called? Myelin. Yeah, those are myelin sheaths. Good. Okay, so let's go ahead and move him aside, and I'll talk about this. I'm looking at the neuron. I said here's the cell nucleus, and let's label the outside first. So this guy here, these things sticking out. Does anybody remember what I just called those? Dendrites, right. These are your dendrites. Okay. Moving on down from your dendrites, you have this part of the neuron here. What part of the neuron is that? It contains like the nucleus and that's your cell body or your soma. Yeah. So let's label that. That's your neuronal cell body. Another word for that is soma. Okay. Cool. And then moving on down the chain, we have a special part that connects the cell body to this long thing down here. And that exists right in here. Yeah, that's the axon hillock. We'll talk about the importance of the axon hillock here in a little bit, but you should know that the axon hillock is really important for reaching threshold. We'll talk about action potentials here in a minute, but that's your axon hillock. Okay, so dendrites receive information on the outside. You have your soma or cell body, and then you have your axon hillock, and then on the way down, you have your axon proper. So this is going to be your axon. And then at the very base, we have what's called the axon terminal. Okay, all the way down. Can you see that? All the way down. At the very end of the axon, there's something called the axon terminal. And then there's going to be this space. So this space, I'm just going to draw here in green, right in here. It's all this space. It's the space between this first axon and this second axon. Okay? I'm just going to label that as space. And that space has a special name, and it's called the synaptic cleft. Okay, so basic parts of the neuron. You have your dendrite that's attached to your cell body. And then you have your axon hillock, which attaches your cell body to your axon. Long strand all the way down is going to be your axon. At the very end of your axon, you have your axon terminal. And the space in between this axon and this axon is called your synaptic cleft. Now, since we have this, the flow of information is going from here down through to the next neuron. That's the flow of information. So this neuron comes first. So this is going to be known as the presynaptic neuron. So here I have axon terminal. I'll just go ahead and add presynaptic. There's your presynaptic axon terminal because it's in front of the synapse, which makes these things on this axon postsynaptic. So this is going to be your postsynaptic dendrite. So same thing here. So you have your dendrites, your cell body, axon hillock, axon terminal. But this is your presynaptic neuron, and that's your postsynaptic neuron. So essentially, something funky has to happen to transfer information from the presynaptic neuron to the postsynaptic neuron through the synaptic cleft. Questions? Everybody got that? Okay, let's go ahead and think about the flow of information going in this direction. Now, what happens to allow the flow of information? So information is received here in the dendrites. So let's say it's my finger touching the stuffy. I can feel that or not feel that. What happens if I feel it? Does anybody know the word? Action potential, right. Yeah, so let's talk about an action potential. That's going to be the next part. Have you covered action potential yet in physiology or?
Not here. Yeah.
Negative 70.
Negative 70. Okay, negative 70 is going to be resting membrane potential. Okay, usually, so where you haven't covered this yet in physiology.
Not.
Like touch on it about like little by little.
Okay,
Yeah.
but not. Have you covered it? No, you haven't covered it yet, but you're going to get to covering it. Oh, usually it's covered by the time I get to this point, but okay, that's fine.
We'll do better.
Yeah, so your action potential is what happens to allow the transmission of the signal. It's an electrical signal down from the dendrite all the way through the axon terminal. Okay, nothing more than action potential. I mean, if you've heard this before, that's great. Most times people have heard. Has anyone never heard of an action potential before? Okay. Yes? You have. Good. All right. As long as you have. That's great. All right. Let's go ahead and start then. We know that minus 70. This is the membrane potential in millivolts. We're going to start here at minus 70. That's going to be your resting membrane potential. Having said that, does anybody know what minus 50 would be? Yeah, that's going to be your threshold. Good. And then we're going to come back down, and then that's on the y-axis, and looking at the x-axis here, we're just looking at over time. Okay, what happens over time to allow the flow of electrical information to flow down one axon? Okay, so we start out here. Let's start out at minus 70, and I'm just going to go like this, and I'm going to label it with a 1, and then, see that? Number 1 is going to be minus 70 is resting membrane potential. Okay, that's where we're starting. And there's a lot to action potentials. For the purpose of this information, I'm just going to teach you what you need to know to understand neuroscience. That's it. Electrochemical signal. Okay. Now, something happens here. I'm going to receive some sort of stimulus. Let's just say stimulus from the environment. So for the sake of argument, I'm just going to say, you know, I'm trying to touch my stuffy, but I don't really feel it yet. I mean, I'm almost there, so it's going to be something called a graded potential. Okay, this is going to be a graded potential. Okay, that means that there wasn't enough of a stimulus there for me to trigger an action potential. Another way to think about it is like if you have a pin and you just touch your finger or you touch some part of your body. Okay, it doesn't really hurt yet. There's a point at which it hurts, right? So you can put, when you push it in, it's like ow. Okay, that ow, that means you've got to threshold, right? That's another way to think about it. So if I don't quite get to threshold yet, that's what this indicates here. Okay, I'm not quite there. Started at minus 70. I feel a little bit of something. Okay, but I didn't quite make it to minus 50. Okay, but this thing has to happen first before we can get to threshold. And that's going to be a local potential. The local potential has to happen first in order to trigger the action potential. And your local potential is mechanically gated. Okay, there's a mechanism, whether it's going to be pain or touch or vibration or proprioception. It's going to be mechanically gated or graded potential. Okay, that's number 2. Okay, let's say then I come back down to my resting membrane potential because we didn't quite reach action potential. And this time I have a pin and I'm going to poke a little harder, and this time I feel it. So once I hit, that feeling indicates that I've reached threshold. Okay, and I'm going to go, once I reach threshold here, that's when the action potential happens, and it's an all or none thing. Once I reach threshold, I'm going to go. Okay, with the mechanically graded potential, I don't necessarily have to fire all the way. I can perceive a little bit of something, but I can back right back down. But with the action potential, once I reach threshold, it goes. Okay, and that going indicates transmission of information down the axon itself. Okay, so number 3 here is going to be threshold. Okay, and at once I reach threshold, then voltage gated sodium channels open. And once these sodium channels open, sodium rushes in, and the cell, sodium's a positive charge mineral, you're going to come up and become very much more positive. Okay, and this quick change here means that your sodium channels are open. And sodium is going to rush into the cell from the extracellular environment. Okay. At my peak here at 4, what happens is that the sodium channels close and my potassium channels open. So at this point, sodium has rushed in, but my potassium channels open. When my potassium channels open, potassium is going to leave the cell and go into the extracellular environment. So here is where potassium channels open. Okay, so as potassium starts to leave the cell, then the cell itself becomes more negative in charge. Okay, and you come down here, and as the potassium channels continue to stay open, the sodium channels are closed, you're going to get a little bit hyperpolarized. So you're going to come back down to your resting membrane potential, and then you're going to have a little bit, potassium channels still stay open a little bit longer before they close. And then you return back to resting membrane potential. Okay, this little dip here is just a hyperpolarization. Okay, so essentially, in response to a mechanically graded potential, you get a voltage gated potential. That's the action potential, where your sodium channels open, reach a peak, and then potassium channels open, come back down, and then you actually hyperpolarize. Okay, so this is a depolarization, this is a hyperpolarization with the action potential. Questions? Hopefully. Hannah?
Is hyperpolarization a step, or is it just like?
It's part of the process. So, okay. Another way to think about it is if let me go to one more slide before I answer your question. Okay. It is a step in the process, but it also indicates if something is hyperpolarized, the chance of it reaching threshold is much less than it was here. Okay. So if I'm here versus here, this requires a much greater stimulus to reach threshold than this does to get to here. Does that make sense? So I think about depolarization meaning something's going to fire. Think of hyperpolarization as something isn't going to fire. So there's going to be a lack of a signal. to turn the neuron on. Okay, lack of signal transmission. Yes, Mackie? Yes, so the sodium channel is closed. Okay, so at step number 4, I wrote it over here, but I didn't write it up here. So step number 4 is written down here. So here the sodium channels close and then the potassium channels open. Okay. Can you see that now? Okay. The potassium channels close. I'll put that down here. At the peak of hyperpolarization, we'll label that here. Okay. Let's say here is where your potassium channels close. Okay, so you're hyperpolarized and then you slowly make your way back up to resting membrane potential. Okay, so the action potential itself is a depolarization, and then the hyperpolarization is the opposite. Okay, so something's going to fire, it's going to depolarize. Hyperpolarize means that it's way away from transmitting a voltage potential. Questions about that? Is everybody? Hopefully that's a review, and I do know that Dr. Blair will go into much more detail, but this is important for what you need to know to understand neuroscience kinds of stuff, and we'll get to that now. Let's look at this next one. It's called synaptic communication. Here, hopefully where you'll see a little bit more clearly what I was trying to show before. Kind of the same picture that when we look at parts of the neuron. So we can just go ahead and quickly label this part up here. What's this called? Dendrite. Okay, and then I don't remember which color I used, but let's go ahead and make this the soma. Okay, and then underneath the soma we have this area here, which is the axon hillock. Okay. Okay, so there's a resting membrane potential. So let's just say that this neuron is at resting membrane potential. So he's at minus 70 millivolts. Okay, let's say there's some sort of stimulus, some sort of mechanically graded stimulus. So if you want to consider pain, I can finally feel the pinprick, okay? As I feel that pinprick, what happens is that sodium starts to rush into the cell at threshold. Let's label that as threshold. At that mechanical threshold, I can feel it, so sodium starts to rush into the cell. When that cell gets to the point where you reach minus 50 millivolts, then when that minus 50 millivolts hits this axon hillock, the action potential happens. Okay, and the action potential itself is nothing more than the transmission of and the opening of sodium channels. This happens all the way down the axon. So this potential, this action potential happens here, transmitted through the axon hillock, and happens all the way down through the axon. So a series of action potentials. So everything's firing. Okay. As it continues to fire all the way down to the axon terminal. So let's just, these plus signs just indicate that an action potential has happened. So this is your action potential. to more than electrical chemical signal, all the way down to the bottom, and then we get to the axon terminal here. This is your, once the action potential reaches the axon terminal, in this case the presynaptic axon terminal, something different happens. When that action potential reaches the axon terminal, does anyone know what happens? Yeah, but something happens to cause the release of neurotransmitters. Something happens. So here what happens is, I'll put it in orange. What happens is that a special kind of channel opens. So this is, I'm going to represent, this is a calcium channel. So calcium channels open and calcium comes into the cell. Okay, so here, let's put it here, calcium channels open. So there's an electrical stimulus traveling down the first neuron. When that electrical stimulus hits the end, or the axon terminal here at the bottom, it causes calcium channels to open. Calcium comes into the cell, and what happens when calcium comes into the cell is that this causes the release of special chemicals called neurotransmitters out into the synaptic cleft. Okay, I'll just label this as, these are my neurotransmitters. So the transmission of an electrical signal down one axon to indicate that threshold was reached is now changed to a chemical signal by the release of neurotransmitters, and it's that chemical signal, the neurotransmitters, that then crosses the synaptic cleft and binds to the dendrite of the postsynaptic neuron. Okay. So what that looks like, let's say that each of these dendrites have some sort of receptor on them. Okay, and when that neurotransmitter floats over and binds to the receptor, it causes another action potential to happen for this neuron. So that's how information is transmitted from one neuron, the presynaptic neuron, to the postsynaptic neuron. We go from an electrical potential to a chemical messenger system via the use of neurotransmitters. Label that as a cell surface receptor. So, as far as neurotransmitters are concerned, does anybody know any neurotransmitters? Sorry? Is acetylcholine? Sure, acetylcholine. Yeah, okay, dopamine's a good one. So let's go with general neurotransmitters. Acetylcholine is going to be a big one. And dopamine is going to be another one. It's a general neurotransmitter we're going to get to when we get to Parkinson's. Acetylcholine is excitatory for the most part, and then dopamine can either be excitatory or inhibitory. And now there's going to be other neurotransmitters. Does anyone know that, so these are two general neurotransmitters, so that there's a major excitatory, no, not serotonin. Anybody? So it's amino acid, glutamate is going to be your major excitatory neurotransmitter, and there's also a major inhibitory Yeah. And that's going to be GABA. G-A-B-A. Okay. So general neurotransmitters, acetylcholine, dopamine, we're going to talk about a whole lot more later on. But you should know that your major excitatory neurotransmitter is glutamate, and your major inhibitory neurotransmitter is going to be GABA, G-A-B-A. Okay. Questions? Where we are so far? We're gonna, yeah, go ahead, Josh.
Does that say cell surface?
Receptor, yeah. So essentially, the neurotransmitters are released from the presynaptic neuron, and they cross the synaptic cleft and bind to the cell receptor on the dendrite of the postsynaptic neuron. This results in the transmission of information from one neuron to the next. Okay, so we went from an electrical signal to a chemical signal. Yes?
And it's headed to the little, like, end of the dendrite?
It's headed to the dendrite. The dendrite's the one that's going to receive that information. Yeah, so it's not necessarily the end. It's anywhere on the dendrite.
Are neurotransmitters kind of like locked key proteins, where they have to be a certain shape?
Mm-hmm. Sure. Yeah. Mm-hmm. Yeah, so anything that's going to bind to a receptor has got to have a specific shape to bind to that receptor to be able to fit into it. Kind of like enzymes in general. Any other questions? Okay, let's take a 10-minute break. So 1 minute after 10.